Hi guys, Miss Francis here. Today I would like to revisit the endomembrane system and get more detailed into some of the organelles that are part of the endomembrane system. But first let's refresh our memory and discuss which organelles are in fact part of the endomembrane system. Well, it includes the nuclear envelope, the endoplasmic reticulum, both smooth and rough, Golgi apparatus, lysosomes, vacuoles, and the plasma membrane. So let's first talk about the endoplasmic reticulum. The endoplasmic reticulum is the membrane factory, where it is a network of tubes known as the cisternae that have fluid-filled spaces. The ER membrane is continuous with the nuclear envelope and then is going to extend at times to the plasma membrane. And there are two types of ER. You're going to have your rough ER and your smooth ER. The smooth ER lacks ribosomes, whereas the rough ER contains bound ribosomes. And we know that ribosomes is the site of protein synthesis. So if you were given an image, would you be able to spot the difference? Would you be able to know that this is the rough ER and this is the smooth ER? Because in the rough ER, you'll see that there are bound ribosomes, whereas the smooth ER doesn't have those bound ribosomes. Enzymes within the smooth ER are also responsible for synthesizing lipids, which include oils, phospholipids, and steroids. And one of the steroids is cholesterol, and that's going to play a role in our phospholipid bilayer. So essentially, the smooth ER helps make plasma membranes. Other functions of the smooth ER include detoxifying drugs and poisons, such as the smooth ER of the liver, as well as breaking down stored glycogen into glucose. Here's a nice little summary of the rough endoplasmic reticulum, some more information, and a comparison. What is the difference between the smooth and the rough? Well, the smooth lacks ribosomes, whereas the rough ER has bound ribosomes, and their jobs differ. Because the rough ER has ribosomes, it's going to make proteins, whereas the smooth ER that lacks ribosomes is going to make your oils, your phospholipids, and your steroids. It's going to detoxify drugs and poisons, specifically the smooth ER that's found in the liver, and it's going to break down stored glycogen into glucose. So here's a summary of what I just said. The ER is a membrane factory. If I were to label the parts, would you be able to follow along and label the parts with me? So let's start by labeling the nucleus. So this entire thing is the nucleus. Would you be able to locate the nuclear membrane? The nuclear membrane is what surrounds the nucleus. Another name for it is the nuclear envelope. Would you be able to tell me that this is the rough ER because of all of the dots that you see, those dots being the bound ribosomes? Here is an enhanced image. It's focused specifically on the rough ER. It's got a higher magnification. And now let's move on to the Golgi apparatus, right? So the Golgi apparatus is what finishes, sorts, and ships cells. It's like the center of manufacturing. It sorts and then ships out the protein to where it needs to go. So essentially, it's kind of like a post office. And it kind of looks like a stack of pancakes where those membranous sacs are known as cisternae. So one side of the Golgi, the cis side, is going to receive material, whereas the trans side is going to send them off to where they need to do their job. This video here is a great video that actually shows the movement of the Golgi body and how it operates. So during their transit, products from the ER are then modified to reach their final state. And I should say during their transit within the Golgi body, right? The products, which include the proteins from the ER, are modified within the ER. Those modifications include the addition of oligosaccharides on the glycoproteins. When we do the cell membrane, we'll talk in greater detail about oligosaccharides and glycoproteins. And then finally, once the appropriate oligosaccharide is attached, or once the appropriate modification has been made to the protein, the Golgi is going to package it into a transport vesicle. Here's a nice little summary. Sometimes you'll see artist's rendition pictures of the Golgi. Sometimes you'll be presented with an actual micrograph of the Golgi apparatus. The next organelle I'd like to discuss are lysosomes. Lysosomes are digestive enzymes. They contain your hydrolytic enzymes, and they're responsible for digesting macromolecules. So do you see I've got two lysosomes, one here and one here? This is my Golgi apparatus that we just discussed. Here I have mitochondria, which we talked about yesterday, and here is another image of my lysosome. So the last image was maybe magnified greater than this image. But basically, the lysosomes can digest proteins, it can digest fats, it can digest polysaccharides, it can digest nucleic acids. Works best at a pH of 5. And sometimes it's known as the suicide sac because a massive leakage of enzymes from the lysosomes can destroy a cell by autodigestion. Furthermore, the leakage of enzymes from the lysosome can also digest defunct organelles. So the lysosomal enzymes and membrane are synthesized by the rough ER and then transferred to the Golgi, and at least some of the lysosome buds from that trans face of the Golgi. Well, then the lysosomes would fuse with food vacuoles that enter the cell via phagocytosis, and as the lysosome digests the polymers, the building blocks, right, the monomers, the amino acids, for example, when we're talking about proteins, pass out of the cytoplasm and become nutrients of the cell. And like we already mentioned, lysosomes can also fuse with another organelle to digest worn out organelles. Therefore, the lysosomes play a critical role in the programmed destruction of cells in multicellular organisms. Sometimes it's known as apoptosis. Sometimes I call it apoptosis, tomato, tomato, potato, potato. But anyway, Tay-Sachs disease actually is linked to the lysosome because it's due to the lack of the lipid digesting enzyme in the lysosome. If you're interested, this is a short video on lysosomes. Here's a summary slide on lysosomes, as well as this slide summarize the lysosomes. Lastly, let's talk about vacuoles and vesicles. So vacuoles have diverse functions. The first vacuole that we're going to encounter is a food vacuole, and the food vacuole would enter the cell doing this process called phagocytosis. The food vacuole would fuse with the lysosome would digest the food that's inside. Contractile vacuoles are found in freshwater protists, and they're going to pump excess water out of the cell. Central vacuoles, those are the vacuoles that are going to be found in many mature plants. They provide turgor. Most of them are filled with water. Where the membrane that surrounds that central vacuole is known as the tonoplast, and it's selective in its transport of solutes into that central vacuole. So here's the artist's rendition of a chloroplast and that central vacuole, and now here is the actual image of the chloroplast, of the central vacuole, sorry. Again, artist rendition and actual pictures showing plant cells. So here's a nice little summary slide about vacuoles. And now let's get to vesicles. Vesicles have a single membrane. They contain fluid inside. They are very small, and they're going to transport materials inside of a cell. If you're interested in revisiting the endomembrane system, this is a nice animation that summarizes all of the organelles that we just discussed and how those organelles work together. Next video, I'm going to discuss with you peroxisomes and cytoskeleton.